Hello everyone, welcome back to another Camber video. Today we are going to be talking about mirror life. You've probably seen articles recently in the past year or two circulating online talking about mirror life and labeling it as potentially the next big doomsday event, which, while this trick works wonders to further stress out and draw attention from a lay audience, I'm not fully convinced that this is a pressing issue as of yet. Now, as a caveat, there is much more nuance to be added to that take of whether mirror life is dangerous or not, and I'll discuss that at greater length towards the end of this video. However, I think that news sources, at least for the most part currently, are trying to distort how scared you should truly be about the potential presence of mirror bacteria. However, what news sources do get right in this case is that we should definitely keep our eye on this field of research into mirror life and start thinking about potential regulations or international agreements so that we have a common understanding for best practices and how to approach this new cutting edge, I guess, type of biological work that is just starting to come out now. To understand what mirror life is, I'm going to take you guys through a brief tour of some chemistry so that you guys can understand the basic principles of what mirror life actually entails and what that looks like at the molecular level. Next, I'll give you a more lighthearted example for some of the interesting potential consequences of mirror life and what it would be like interacting with mirror life by considering what it would be like eating a steak from a hypothetical mirror cow. Finally, I'm going to wrap up this video talking about the potential dangers of this new emerging technology that was just still thought of as a thought experiment until a couple years ago. However, we are at a point where we will soon be able to create the long lost cousin of all existing life today. So join me in today's video where we will cover all things mirror life. So what does mirror life actually mean or entail? Well, when you look at the biological macromolecules that make up all of everything in your cells, there are four different categories of these essential macromolecules that make up everything you know and love. Those are proteins, lipids, carbs, and DNA/RNA. These four together in varying quantities make up the singular cells, of which you have many billions, trillions in your body. And of these categories, these four different categories of macromolecules, almost all of them contain chiral molecules. Now, chirality is a way that chemists determine identical molecules from one another. So I'll give you an example. When we're looking at this molecule in red here, you'll see that there's an intersection of four lines leading to a middle point. This middle point, although it isn't drawn, there is a carbon in the middle there. Carbon notoriously can make four different bonds, and here we can see that denoted through these four different lines that are connecting to it in the middle. So the two straight lines which are connecting to the carbon are bonds which are shown to be in the plane of the page. So you see a methyl group, which is the CH3 up top, and then you also see a chlorine bonded to the center carbon. There are also two other bonds, one with a filled in line, which that represents an alcohol or OH group, and the filled in line denotes that this bond is coming out of the page towards you, whereas the dashed line represents a hydrogen which is coming out of the back of the page, which is bonded to the carbon. So the notation with using straight, filled in, and dotted lines are basically a tool that chemists use to see the three-dimensional nature of molecules on their two-dimensional paper. Crucially, when a carbon is bound to four unique different groups, that makes that carbon a chiral center. When you have a chiral center, that means that a mirror image of that molecule also exists, made up of the same exact elements, but in a different configuration, making it such that mirror molecule is in and of itself also a unique chemical that you need to take into account. Looking at the mirror image of the red molecule we were just looking at, what you can see is if you take that mirror image of it and try and superimpose or rotate that mirror molecule by 180 degrees to match the molecule on the other side of the mirror, you see that the red original molecule and the blue mirror rotated molecule, you can see that they don't overlap, and the alcohol group and the hydrogen are in the wrong spots, meaning that the mirror image of the red molecule creates a molecule which, while it's composed of the same exact elements, they are in a different configuration, making it such that the mirror molecule is unique in its own way and has its own spin, so to speak. The relationship between the red and blue molecule is that they are enantiomers. Enantiomers are mirror images of one another, and they are labeled with S and R spin configurations, which are determined by the order of priority of the elements. However, this isn't a chemistry tutorial, so I won't talk to you about how to determine R and S spins. All that you need to know is that when you have some molecule, you can have a mirror image version of it. Another great way to think about chirality or enantiomers is that your hands are enantiomers of each other. So if you look at your hands, right, if you were to suppose there's a mirror right here in between them, and imagine I'm also not holding the microphone, you'd see that my left and my right hand are perfect mirror images of each other. And if I were to try and put my left hand on top of my right hand so that it matches, you can see obviously they are not the same thing, even though all of them have the same 5 fingers attached to my hand. So another, I guess, quick thought experiment you can do is, let's imagine that you somehow happen to hurt your right hand and you need to get a hand guard to prevent your hand from moving. Assuming you don't get something that is malleable and can mold to both of your arms and you have a more of a fixed, rigid hand guard, then if you were to get a right-handed handguard for your right hand, then imagine that that handguard has a specific R spin or right hand, I guess. And then if you hurt your hand at a later date, you can't use the R handguard, the right spin handguard. You're going to have to go back to the store and buy a new S or left-handed handguard to protect your arm. A specific notation of this spin or of notating mirror molecules isn't super essential to understand. All that I need you to know before we move on is that of the 4 classes of biological macromolecules, almost all of them are made up of chiral molecules, meaning all of them have a mirror counterpart to them. So, in other words, life as we know it today only uses quote unquote left handed molecules, whereas hypothetical mirror life would be using entirely quote unquote right handed molecules. Digging specifically into the 4 classes of macromolecules, let's first look at proteins, which proteins are made up of the same 20 amino acids put together in various combinations. For example, if we're looking at this taste receptor here, which helps you taste food whenever you're eating, it is made up of only 20 canonical amino acids, which are put together in a very specific order to make up that protein structure of the taste receptor. Among being able to create taste receptors, proteins also make up your muscles, transport cargo across cell membranes. They let you make RNA from DNA. They catalyze chemical reactions such as enzymes, and there are many other functions of proteins, and they're all made from the same 20 amino acids. Looking at the 20 amino acids, you can see that all of them have a 1 letter shorthand for them. And all of these amino acids, with the exception of glycine and cysteine, have an S chirality. And these 18 S spin amino acids with cysteine, which is R, it's a rare exception, as well as glycine, which isn't chiral, these group of amino acids make up the so-called L family of amino acids. And these amino acids, the L family, adds up to make all of the proteins that have ever been known to all of life on this planet. All proteins and life everywhere are made up of L amino acids. So a large sequence of amino acids can be put together to make a protein like a taste receptor. Looking at the specifics of this taste receptor, you can see that it has over 800 amino acids put together in a line to make one of the two chains that make up the receptor itself. Zooming in, you can see the exact sequence of this receptor. So, if we had a programmable cell like E. coli, for example, or quote unquote programmable, we could import this sequence into the E. coli and tell it we want you to make this exact sequence, and E. coli could, in theory, produce this taste receptor for us if we wanted E. coli to make it for us, because E. coli, similar to humans and all other life on this planet, also uses left-handed amino acids. Now, looking at the finished product here, we have a taste receptor which is made up of two 800 amino acid long proteins which together interact to respond to the changing chemical environment inside of your mouth. Similarly, carbohydrates, which are made up of monomers of various sugars, are also chiral molecules for the most part. I won't explain this in depth, but just know that sugars, like amino acids, almost all of them are chiral molecules as well. Unlike amino acids, life chose to use D sugars instead of L sugars to draw its energy from the environment. Then, if we move on to lipids or fatty acids, very basic fatty acids aren't chiral. However, when you start thinking about more complex lipids, which are characteristic of modern eukaryotic life, those are chiral. Then there's DNA, which, while nitrogenous bases themselves aren't chiral, these nitrogenous bases attach themselves to D sugars, which then also attach to a phosphate group to make up a nucleotide. And then these nucleotides then add up to contain all the genetic information in your body and make a characteristic right-handed spiral for DNA. Now, if you just for a second imagine that the DNA was, for example, a spiral staircase, then when you would walk up the DNA, your right hand, and I'm doing this opposite because the camera's going to invert it. If you have your right hand and you're walking up the DNA, your right hand will be gripping the outside railing, and as you're walking up, your right hand will be grasping the outside railing as you walk up the DNA. However, in a hypothetical left-handed DNA, which would be characteristic of what mirror life would use to store genetic information, it would be made up of the nitrogenous base binding to an L sugar and a phosphate group. And then necessarily, since you're using the different family, the opposite family of sugars than you are in real life, that means when these nucleotides stack up on each other, then if you were to walk up the DNA of mirror life, you'd be using your left hand and you'd be gripping the outside railing with your right hand as you walk up it, as opposed to the right hand, which is what classical DNA and RNA, the way that it has spun on this planet since the inception of life. So, if we consider life today, which uses L, or let's call it left handed amino acids, and D, or right handed sugars, which make up right handed DNA, mirror life would then sensically have to be life that is made up of D amino acids and L sugars, which would then create a left handed um, DNA. It would be in every which way different to life, just in the sense that it would be the mirror image. The actual sequences likely wouldn't change all that much for mirror life. You would just need them to be the exact mirror image of the sequences of the proteins that your body uses right now. Now, this brings up an interesting question as to why life is chiral at all to begin with, and why life chose to use only left handed amino acids and right handed sugars. Now, it makes sense that once there was a clear favorite, life stuck to either left handed or the right handed family, but not both. See, at the molecular level, if you ever need to catalyze a reaction by using an enzyme, the chemical needs to fit into the active site of these enzymes very specifically to catalyze the reaction of that specific chemical you want to have undergo a reaction. Instead of creating two copies of an enzyme that performs the same function, primitive life likely would have preferred to use the energy which it could have used to make a mirror image enzyme to instead use that energy for survival and instead just focus on having one of the mirror or one of the enantiomers, so to speak, of that enzyme. So this means that there came a time at two different points in the evolution of life where life clearly chose a winner among the L and D families, choosing L amino acids and D sugars, and that choice has stuck with us ever since that moment. It isn't quite clear yet why life chose the way that it did for using the L and D families for all life's macromolecules. And if we wanted to have a better explanation as to why this might have happened, we would have to consult the field of abiogenesis, which studies basically how life started on Earth. But this is a very dense field that perhaps I'll cover in the future. But for right now, all I can tell you with certainty is that life just prefers certain macromolecules over others, and that is just the way it is. And we aren't really sure why it's L amino acids over D amino acids and why it's D sugars over L sugars. That's just what life chose, and that's just kind of what we have to roll with. So, what would it be like to actually interact with mirror life? Well, as I mentioned earlier, many researchers are thinking about the possibility of mirror life overrunning our world and mirror bacteria potentially being extremely lethal and having no cures, so on and so forth. But before we delve into that discussion of the danger of mirror life, let's consider what it would be like if we could potentially eat a steak from a quote unquote mirror cow. So this obviously isn't possible to test in reality, but just as a fun thought experiment, let's think about what it would be like to taste a steak which is made from all of the mirror counterparts to what a regular steak would taste like. And can we consider, would potentially a mirror steak be a lot more nutritious or beneficial for us than a regular steak? So if you were able to raise a cow that in all the cells and everywhere all across its body was using right-handed amino acids and left-handed sugars, then if you were to use that quote unquote mirror cow to make a steak, you would have a mirror steak. Now, the thing about a mirror steak is that likely it would taste wildly differently than the regular steak we are used to. The reason for that is that your flavor and olfactory slash smell receptors are proteins themselves made up of their chiral L family amino acids. And these amino acids, which make up that flavor receptor, are used to binding life compounds, not their mirror life counterparts. This means that while taste receptors can bind both life and mirror life counterparts are rarely found in nature at all. Importantly, mirror compounds do also are able also to bind these taste receptors. However, they have radically different taste profiles than that of the mirror image that you would find commonly among nature. For example, one of such, I guess, mirror compounds which have a radically different taste profile is that of carvone. The taste receptors in your body are used to binding S-carvone, which is a molecule typically found in a steak. And this is a chiral molecule which smells like caraway seeds slash rye. Now, this taste receptor, if instead of binding to S-carvone, which has a rye caraway seed taste to it, if it were to instead bind R-carvone, which is the mirror life counterpart, then interestingly, R-carvone is known to have a distinctive spearmint-like taste to it. In fact, something else that would be different about the steak is that it would very likely be missing the characteristic umami flavor of steak, which typically is associated with the interaction of L-glutamine with your taste receptors. D-glutamine, on the other hand, which is the mirror life counterpart, is reportedly much more muted flavor wise comparatively, and in general, the whole family of D amino acids have a much sweeter taste to us than L family amino acids do, which are generally more savory. Interestingly, something that would stay pretty consistent between the mirror life steak and a regular steak is the saltiness or sourness of the meat. And the reason for this is that the receptors which create the sour and salty sensation in our mouth are interacting with hydrogen ions or protons and sodium ions, respectively, for creating that sour or salty sensation in our mouth. And since hydrogen ions and sodium ions aren't actually chiral, that means that this part of our taste would likely still be intact when eating a mirror steak. Another interesting thing to consider is that since all of the protein in this steak is the mirror image of itself, despite the fact that it is all a mirror image, it likely would have the same consistency or bite or chew to it that a regular steak would, because there is no reason to believe that the mirror image counterpart of proteins would not have the same characteristics, texture wise at least, that we would get from eating a regular steak. So the bite to it, I guess, would also be conserved in a mirror steak. However, the last interesting thing about this steak is that while you could eat it and it would be filled like chock full with protein, the thing is your body wouldn't actually be able to digest any of the right-handed protein, quote unquote, in this steak. So since all of the enzymes in the body are used to cutting L or left-handed amino acids, that means that all of the protein would essentially flow right through you and you would have no nutritious benefit from all of the abundant mirror protein in the mirror steak. Similarly, all of the left-handed sugars in this steak would not be able to be transported into your cells and used to create energy for your cells. The reason that they wouldn't be able to do so is because sugar and amino acid transporters, they are a lot less promiscuous, or as in they don't bind as much stuff as carelessly as taste receptors do. Taste receptors love to sample all of the compounds in the environment, whereas a transporter, which is taking sugar or amino acids from outside the cell to inside so it could be processed for energy, are much more stringent on what it lets into the cell, and likely the mirror image counterpart of sugars wouldn't be able to cross into the cell and be used to create energy. This is a similar principle to how aspartame works, artificial sweetener, because aspartame, it can bind the more promiscuous taste receptors which don't really care as much about what they bind. They're very happy to bind aspartame, but then when aspartame actually starts flowing throughout your body, none of the transporters of the cells within your body are actually willing and happy to take aspartame into the cell so that it can be used for energy. And that is how you can taste the sweetness of aspartame but not actually get any of the benefits of the energy that the I guess aspartame has locked within it as chemical energy. So just summarizing then, if you were to eat a mirror steak, it likely would taste a lot sweeter, potentially even radically different than a regular steak. It would have the same chew or mouthfeel to it, as well as some of the sour and salty taste to it. But at the end of the day, it would taste pretty differently, as well as you probably wouldn't be able to get any nutritional benefit from this steak, which, as you guys know, is very rich with macronutrients. So if you were to somehow able to mass produce mirror steak, you would be able to potentially have, I guess, a steak which tastes a little bit like toothpaste potentially, which doesn't give you a lot of energy but still has the same bite or chew to it that regular steak does. And just to clarify, this is a simple thought experiment, and I'm in no way saying that this is 100% what a mirror steak would taste like and what that experience would be like. I just think that it is funny to think about if you were to bite into a steak that it would just taste like toothpaste and maybe a little weird saltiness to it. And I just thought it was a fun example to bring up before we get bogged down now on talking about, is mirror life actually dangerous to us? And to just give you the down low up front, should you be worried about mirror life as the next potential cataclysmic doomsday event? I would say don't worry about it. I think that there is some danger to be had in studying mirror life. However, I think there's a lot more dangerous things than mirror life currently. For example, the current pace of AI development. However, let's try and discuss some of the dangerous factors that scientists are saying mirror life could be potentially life threatening towards the human race. One of the main papers that I read to think about some of the dangers potentially present in creating mirror life is an editorial published in Science by Dr. Kate Adamala, along with many other researchers who all were pressing for the need for more regulation and the potential dangers and warning about what creating a mirror life could potentially lead to. One of the main reasons why they believe that mirror life or mirror bacteria specifically could be dangerous is that since this mirror bacteria would be composed of all right handed amino acids and left handed sugars, it could be extremely deadly, seeing as our immune systems are trained for since the beginning of life on Earth to target stuff with left handed amino acids and right handed sugars. This means that it could be likely that should mirror bacteria ever be made, they probably could evade our immune systems very easily since our immune systems aren't trained to be looking out for mirror bacteria because that has never existed here on Earth before. Further, bacteriophages, or phages for short, are viruses which hunt down and solely kill bacterial life and are a way of, I guess, regulating. They are part of the ecosystem that regulates bacterial growth. And interestingly, phages likely also wouldn't be able to interact with mirror bacteria, as since too their defenses are ingrained with life's choice to use left handed amino acids and right handed sugars. It is also worth noting that all antibiotics currently in use today would also likely be futile to stunt mirror bacteria growth. Since most antibiotics themselves are chiral molecules which bind to chiral targets such as proteins on bacteria, it means that if you switch the chirality and instead want this antibiotic to bind the mirror image of what it usually binds to, it simply will not be able to do that. This is comparable to the analogy of the handguard that I was talking about before. So, for example, let's say that the my hand here is the drug target, and the cast, or I guess handguard itself, is the drug that binds to the drug target. Now, if for all of the time life has existed, there's left handed life, and we're developing left handed handguards to put a damper on some of the left handed living molecules, if we then have a right handed, I guess, protein suddenly exist, you can't use the same left handed handguard on the right handed life molecule. And that is basically a very simplistic way of saying we would have to start from ground zero making antibiotics for mirror life should they become deadly to us. One thing to keep in mind, though, about mirror bacteria is that all of life today uses macromolecules, which mirror life likely wouldn't be able to digest or gain any energy from in any meaningful way. Alongside that, if you have mirror bacteria, it would have proteins which classically normal bacteria would use to try and get into the cell. Those proteins in the mirror bacteria, which are used to infiltrate the cell and replicate, likely might also not work since you have the same issue of using a right handed protein to try and interact with a left handed protein to get into the cell. So, that is something also to mention as well. But a big crucial aspect of that is that even if it could get into the cell, firstly, the mirror bacteria would need to find a way to eat regular life macromolecules because it would be used to processing mirror, I guess, compounds to produce energy. So, in the same way that I could eat a mirror steak and there'd be very few molecules in there that aren't chiral that I would be able to actually digest to give me energy, likewise for the mirror bacteria, if it does go unnoticed by my immune system and somehow gets into my cell and then starts looking around for any goodies in there, there's a chance that its protein machinery wouldn't be up to the task of digesting all of the stuff in my body, which is the other handedness compared to the mirror bacteria. So it is reasonable to think that if mirror life were to be created, it would be extremely inefficient at first and would be heavily reliant upon researchers for feeding it those mirror compounds which aren't very abundant in nature at any significant capacity. So the one caveat, I guess, counter argument that I made to my own counter argument is that while yes, it is possible that bacteria wouldn't be able to even get in your cells and possibly couldn't even be able to eat the food that your own cells eat, bacteria notoriously evolve way quicker and on much shorter timescales than humans are able to. So, for example, if we think about the famous Lenski long-term evolution experiment, researchers started feeding citrate to E. coli. Citrate being a molecule which E. coli are known to not be able to digest. And over the course of this experiment, which started in 1988, we are now at a point where the researchers have created quote unquote synthetic E. coli, which are now able to digest citrate. Now, the reason that this was able to happen was because the way in which the experiment was devised was that it placed immense evolutionary pressure on E. coli to get its energy source from a molecule which classically E. coli is not known to be able to digest, making in essence a synthetic E. coli which eats citrate unlike any other E. coli living on the planet today. So, if we're able to modify E. coli to eat citrate over a couple decades, it isn't too far fetched to think that mirror bacteria could too evolve in a similar way and use current life molecules to feed itself, other than instead of using other handed mirror molecules to feed itself. All of that being said, though, let's look at the most recent advances in the field of synthetic biology to see how close we are actually to making a mirror life bacterial cell. So, we're going to be looking at a preprint that was published to bioRxiv recently by Dr. Kate Adamala, the same professor who wrote the editorial to Science, basically voicing her concern about creating synthetic life. She recently published preprint, um, which I will say, since it is a preprint, just to be clear, this paper has not gone through peer review, and it could be months to years before this paper is actually finalized and published, um, and all of that. So some of the claims in this paper might be walked back or have to be re experimented. But with that disclaimer out of the way, let's just look at this, I guess, new type of synthetic life, which seriously shocked the, I guess, scientific world when it came out just recently. So, what Dr. Adamala's team did is that they created a hybrid genome which contains pre existing genes from E. coli as well as a few genes taken from phages. So, they made basically a basic membrane using simple lipids and then used a genome which had for the most part bacterial DNA with some viral DNA, and they were hoping that those few genes and that membrane was enough to make this, they called it a spud cell. They were hoping that the spud cell would be able to create daughter cells and replicate on its own. Looking at the efficacy of the spud cell and how easily it's able to replicate on its own with this synthetic genome is after 5 replication cycles, they noted that only 30% of spud cells had intact genomes. Now, if we were to compare these numbers to the accuracy of human DNA replication, there isn't even a comparison to be made. If you round to the nearest tenth to the amount of times that replication goes wrong in human cells, you would be at 100% efficiency because there's almost no errors that are made in the replication process compared to what the spud cell is going. So, spud cells are very far from surviving on their own, and in fact, the researchers needed to feed these spud cells on their own. They weren't able to take in energy from their environment, and they also had to input mechanical stress to actually separate the membranes from themselves so that you'd have a basically father cell and a daughter cell to basically replicate the spud cells. So the spud cells weren't able to bud off a cell to replicate either. They required mechanical stress input from the researchers as well. So that alongside having to import many proteins and organelles such as ribosomes into the spud cell to kickstart its ability to quote unquote reproduce on its own was necessary. So if you've heard of ribosomes, they are the cellular machinery that makes up proteins from RNA sequences. And they had to import these ribosomes as well as um, other sorts of compounds from E. coli cells. They had to import them since the spud cells couldn't make it on their own. This means that when these ribosomes or other proteins that they imported from live E. coli, after they stopped doing their job, the spud cell simply died out. So this is no shade to Dr. Adamala's work because I think it truly is beautiful that we've gotten to a point where we are able to make kind of a Frankenstein hybrid of bacterial genes as well as viral genes, and they are enough to create a replicating cell, which, albeit it isn't actually a cell that lives on its own, it requires a lot of care from the researchers to actually let the spud cell replicate. It is beautiful that it is a thing that is currently going on, but I think that you should um like seeing this as the forefront of synthetic biology. I think is pretty clear to say that mirror life is still nowhere near becoming an actual problem. In fact, in the editorial where Dr. Adamala wrote, they come to the conclusion that it is likely that within the next 10 to 30 years, mirror bacteria might become a problem. So, well, as I mentioned earlier, it is right for the news sources to say we should be worried. There is still plenty of time to come up with regulations for this technology and international agreements for how we should move forward in studying these types of, I guess, new serious additions to the field of biology. Bottom line, as I mentioned, I don't think that you should be worried about mirror life. However, this is just my opinion, and as always, I encourage you to engage with this topic on your own time and come to your own conclusions. But personally, I'm not holding my breath for the takeover of mirror bacteria, and if anything, I'm more worried about the current pace of AI development. The closest that we have, as of yet, come to making mirror life is, I guess we could talk about the spud cell as the first case of synthetic life, but we are actually producing mirror peptides or mirror proteins, which are being used therapeutically, which on their own aren't really that dangerous because, um, as I mentioned, like your immune system can't really target it. It's just going to be random junk floating around in your body, but we are making D peptides or right handed peptides and using them for potential drug applications, which I think is a very new and up and coming exciting field in drug development and is something that I think we should actually spend a lot more time looking into. So, an example of this type of research is from Dr. Michael Kay, professor at the University of Utah, who uses D peptides to make anti HIV peptides, which themselves are right handed. Now, since HIV is similar to us, came from life which originated on Earth, because of HIV being from Earth, it uses only left handed peptides similar to, or left handed amino acids similar to the way that we use left handed amino acids. So, since these drugs would be right handed, this means that it would be difficult for HIV to degrade these drugs since it is as foreign to them as it would be to us. Mirror peptides are nowhere near close to actually being the whole shebang of actual mirror life and making a mirror life bacterial cell, but there are many leaps forward in technology, such as being able to make right handed peptides much more regularly now, which make it that mirror life is going to be a prevalent issue in the future. However, as I mentioned earlier, current researchers are thinking that we have 10 to 30 years before that happens. So I'm just here to tell you, I don't think that you should worry. We aren't at risk anytime soon of another apocalyptic event, but you never know. That is just my analysis on this topic. I hope, as always, that you found this video informative and hopefully enlightens you to see how media loves to put a dramatic and hyperbolic spin on scientific discoveries and painting them in a much different light than the way in which scientists will view them. And I hope that you've enjoyed this video. And as always, I will see you in the next one.